We are going today to talk about immune receptors. And immune receptors, if you study in the school, it's the most complex genetic system created by nature. If you look at the slide, I would like to remind you that B-cell receptors, uh, B-cell receptors, T-cell receptors, and antibodies, which are soluble form of B-cell receptors, are generated organized very similar way with CDR3 regions shown in circle, recognize foreign antigens. If you think from biological point of view, it's universal diagnostic system created by nature in order to recognize any foreign antigen or pathological conditions or molecules in your body. And complexity of this system is just enormous. It's at least 100 million of different immune receptors are constantly present in our body, and they organized in generated by genetic recombination from V, D, J, and C regions, rather random way, creating this enormous complexity. The practical question, if you're talking about diagnostic system, how to use this system in order just to identify any pathological conditions or disease in our human bodies. Problem here, why it cannot find broad application, because we don't have really direct link between immune receptor sequence and specific antigens which they recognize. If we solve this problem, in this case, it will be an outstanding really system which can be used in order to diagnose practically most of human diseases. From practical point of view, how to study this immune system? The question is enormous complexity. If you think, what does it mean 100 million of different immune receptors? It's at least 1,000-fold more than the number of the human genes. If you are talking about to apply the same tools as what you use for gene expression profiling, you can see where's the problem, because can we analyze 1,000-fold more complexity than really the number of the human genes? And so today we are going to, in order to facilitate these studies, we developed several technologies like illustrated on the next slides. And first, it's adaptive immune receptor profiling assay, which is allowed to do it in bulk profiling of TCR and BCR clonotypes. And we use clonotypes with the name of specific CDR3 sequence, which corresponds specific clone of B cells or T cell. We also going to talk slightly about single cell air assay for T and B cells sorted in 96-cell play, which allow to get full-length chain pairing information for immune receptors, which are necessary for understanding their function and study their properties. And top biomarkers, about 30 markers for T and B cells. And we also slightly we're going to talk about genome-wide expression profiling, which allow to do it bulk expression profiling of all protein coding genes. And all these technologies are designed based on the multiplex PCR. So that's why they are rather similar in concept, how you're going to use them. And we are going to discuss the application of this technology for screening of activated immune receptors with peptides or vaccines, and how to validate this type of the interesting clonotypes what you identify in the course of these studies using artificial reporter system based on JURCAT and K562 cells. As introduction to the technology, here is the example of the assay what we designed for adaptive immune receptor or AIR assay, profiling assay. And what you can see here is structure of immune receptors. On the top you can see you can use this assay not to amplify from RNA, like shown on the top, or from DNA, like shown at the bottom. CDR3 region shown here in the center, which is, it can be amplified using flanking primers, reverse primers, or forward primers. In this case, you amplify CDR3 region. What's great in this technology, what we developed, you can amplify all 7 chains for all TCR and BCR genes. So it can be alpha, beta, gamma, and delta for T cell receptors and HKL for B cell receptors. So you can start from RNA or DNA, and RNA provide you more sensitivity. DNA give you more quantitative measure of copy number of the T cells or B cells, which has specific CDR3 sequence. You can amplify only CDR3, like shown here on this slide, using combination of internal primers, or you can amplify if you use FR1 and combination with C primer, you can amplify full-length region, which includes all CDR1, 2, and 3 regions of the gene of the immune receptors. What great in this technology, you can not only use RNA or DNA as starting material, you can directly use cell lysate in order to amplify all these molecules. Quantitative analysis facilitated of using validator barcode. And validator barcode is similar concept to universal unique molecule identifier, which allow you to correct number of different mutations introduced in the PCR or next generation sequencing step. And data analysis are integrated with IMXR and platform software package, which is dominating platform right now for immune repertoire profiling. In terms of protocol, here's the illustration of another assay what we developed. It's genome-wide expression profiling for 19,000 protein coding genes, and here number of the primers is significantly higher. We have here 19,000 reverse primers, like shown here on this slide, 19,000 for the forward primers, which is designed to amplify small region between 100 to 250 base pair, or from open reading frame of most human protein coding genes. Sensitivity is higher than RNA-seq technology, and again, it's more simple protocol than RNA-seq because it can start from the total RNA without ribosomal RNA or globin depletion. And the last technology what we are going to introduce today, it's single cell analysis technology. It's some sort of the simple technology which allow you, if you just use, for example, expansion of or stimulation of T cells stimulated by proteins, or B cells stimulated by any proteins, you can sort activated cells using a cell sorter in 96 cell plate and perform both single cell profiling of T cells or B cell receptors at the same time markers. And these cells can be sorted based on the expression of activation markers or staining, for example, with MHC peptide tetramer or any labeled protein. As a result, you can get a sequence of the T cell and B cell receptors and expression of 30 marker genes. And in order not to be boring about talk about technology, let's try to talk about how the data analysis. First question is rather simple. Can we quantitate immune repertoire profiling data? Is it possible or not? If you try to run this experiment, for example, take purified RNA from PBMC or whole blood samples and run assay what I described, you generate approximately 100,000 clonotypes or different CDR3 sequences as illustrated here on this slide. What good here, you have rather balanced number of TCR clonotypes, approximately 100,000, and BCR clonotypes identified using this assay. TRD and TRG, it's gamma and delta chains of T cell receptor, it's very rare, so that's why the number is low. But the question is, first question when you try to run this assay, are these data reproducible or not? And simple answer, it's not. The data are not reproducible. If you try to repeat this experiment in triplicates and compare a set of CDR3 sequences which you identify, you will see that at least 99% of the sequences are 99,000 from 100,000 are amplified only from single molecule, like shown here on the y-axis. You see his starting copy of mRNA molecules used for amplification of these immune receptors. Definitely, if you have one molecule in your RNA sample, it's random choice. You amplify or not amplify. In order to get statistically reliable data, you need to have minimum 10 molecules. So this group of the genes, approximately 1,000, it's really allow you really to do quantitative analysis. And next slide is illustrate. Definitely, if you concentrate it on the top abundant clonotypes which present in your biological sample, in this case, you can get reproducible data, is illustrated here on this slide for the RNA or for DNA. For DNA, definitely require more DNA, which is from practical point of view, it's more complex because not always you have several micrograms of genomic DNA. So RNA will give you opportunity to really generate good reproducible quantitative data using significantly small amount of the RNA between 10 to 100 nanogram if you are talking about blood or other lymphocyte-rich samples. The next question is rather practical. Okay, we identify all these 100,000 clone types. What we are going to do with these guys? Definitely the number one problem in this type of the studies, how to identify clone types which activated by, for example, different disease conditions or drug treatment or, for example, vaccination. And in order just to give you example how it works, this technology, I give you example of vaccination studies. In these vaccination studies, we just collect samples, whole blood samples from triple vaccine, tetanus, diphtheria, and pneumonia, and Shingrix vaccine. We collect multiple time points. It's so classical longitudinal studies where you compare samples before vaccination and after vaccination and go to look at the expression profiling data and identify activated clonotypes. If you try to run driver map genome-wide expression profiling assay, which allow to measure protein coding region in 19,000 protein coding genes, we can see for vaccination 1, after 2 days of vaccination, we can see activation of innate immunity pathways, like shown here. And approximately at 1 week, we have a bunch of the genes which belongs adaptive immune responses. If you look at the same, we run the same samples immune repertoire profiling assay, we generate data like illustrated here on this slide. If you look at the right portion of this slide, which show IgH, so it's heavy chain of immunoglobulin or B cell receptors, you can see if we amplify from DNA, like shown here, at approximately 1 week after vaccination, for both vaccination 1 and vaccination 2, we have strong activation, expansion of the clonotypes for maybe 50, between 20 to 100 clonotypes, which belongs to the B cells, which probably we can speculate it's really proliferation of these clonotypes, which differentiated in plasma cells and start to secrete immunoglobulin G M molecules. The same data can be generated using RNA assay, but you can see immediately on this slide the sensitivity of detection of clonotypes is dramatically more sensitive than using genomic DNA, which is rather easy to explain because plasma cells have activation at least 100-fold of BCR gene in the course of activation. If you look at the T cell receptor, which are 2 sections on the left slide, section of the slide, you can see if you use DNA, we don't see dramatic activation of expansion of any T cells, but we still can see activation of T cells at the level of the transcription. So which means probably that T cells and B cells work cooperatively in this type of the immune responses. Based on this result, we speculate that really it's a rather good idea to run both rDNA and rRNA and normalize data to each other. In this case, you have chance to identify activated clonotypes by comparison these 2 data sets. And maybe for longitudinal studies, which I described, it's not really very relevant because you have multiple time points, but you have only 1 single sample. In this case, it's maybe good strategies to detect activated clonotypes. And next section, okay, we identify activated clonotypes. And typical classical example, similar example, in order to do it right now, it's a lot of studies in order to develop immune cell-based therapies. And next presentation will be describe these approaches in really big details. But what people are doing in these studies, they identify candidate peptides, which, for example, associated with the cancer. And it can be multiple strategies, including experimental, like mass spec identification of immune-specific, cancer-specific peptides eluted from the cancer cells, or prediction of the cancer neoantigens based on the mutation, changes in splicing, or any other biological process. After prediction, you need to screen these peptides, and the best strategy is to screen against PBMC or from normal or disease samples. And as a result, you can develop cancer-specific vaccine or cell therapies. And here's the classical example of how it's done, this experiment. So if you take PBMC from healthy or disease donor sample and treat them with candidate peptides, which you predicted, you, after some stimulation for which can be 10 days or 2 weeks or 3 weeks, you really identify expansion of the clones like shown here on the bottom of the slide. So you see here it's in control, it's practically no expansion, and after 10 days you can see some fraction of the clone types which are expanded. After, usually in this experiment, we are doing restimulation at approximately 10 days or 2 weeks after primary stimulation, and really to identify activated T cells by sorting, for example, in 96 level plate, or using single cell or single cell 10x genomics assay. So we just illustrate that we use a combination of rDNA and RNA assay in order to detect activated and proliferating clone types, and the really single cell assay in order to identify these activated clone types after second stimulation. And you can see after second stimulation, if you compare results, and this one is classical bubble plot where the size of each spot corresponds to the size of clone type, number of the T cells for each specific CDR3 sequence. We can see if we stimulate normal PBMC cells from normal donor with CMV peptide, we can see a bunch of activated clone types like shown here on the bottom slide. Important to mention that data are not perfect, because if we just take control DMSO control, we also can see some sort of activated clone types. The question is how to identify correct clone types from these studies. And in order to do it, really, we believe that it's the best strategy is to look at the overlap between different data set. It can be, for example, rDNA or rRNA, say, or sorting of these cells based on the secretion of activation marker CD137. Overlap between these different data sets can give you really set of candidates of T cell receptors which correspond, which activated by specific peptide. But when you identify this candidate T cell receptor, still it's only beginning of the story. You need still to validate this result. And what we find, we look at many technologies developed right now for validation of interaction between T cell receptors and candidate peptides. And we believe that really one of the best strategies to use artificial reporter cell lines, Jurkat reporter cell lines, in combination with K562 cells. And in this system, which was developed already more than 20 years ago by many researchers, including Rosenberg from NIH, what we are doing here, we just take Jurkat cells like shown here on the left side, and we convert these cells in reporter cell line, where we clone NFAT reporters under control of transcriptional activation promoter, which express GFP if T cell receptor is activated. We knock out in these cells endogenous T cell receptor and introduce CD8 molecules in order to convert Jurkat cells to CD8 cells. So if you clone in this cell, you can clone in these cells individual construct like shown here on the top of this slide, or library of T cell receptors in order to create library of T cell receptors in Jurkat reporter cells. In K562, you can clone any peptide in the context of HLA complex. It can be fusion, like shown here on the top of the slides, which is peptide, beta-2 microglobulin, or HLA, or it can be minigene, which allow you to express on the surface of K562 MHC complex, which will be exposed peptide. If T cell receptor recognize peptide, TCR signaling pathway is activated, and Jurkat reporter cells start to be fluorescent. So this system, it can be used in order to screening individual constructs or libraries against each other. And what we find, the best way to screen this type of the interaction is to use fluorospot microarray array developed by company Celldome with the name Clone Explorer. And this one's a great system. It's based on the using of the microwells, like shown here on the top of the slide. So it's standard 96 well plate. In each well, you have 3,000 microwells. This microwell shown here in details on the left portion of the slide, and you can see here what great in this system, you can monitor in time how many cells you have in each well. how many antigen presenting cells or T cells you download in these microarrays. After that, you can look at the proliferation during the time when they co-incubated to each other. And grade each, like shown here on the right slide, section of the slide, you can see microarrays are coated by antibodies against cytokines. So if any cells activated in this type of the interactions, cytokines secreted and can be immediately detected by staining with the antibodies against these cytokines. So you can see fluorescent signal. In the case of K562 in Jurkat cells, it's more simple because Jurkat cells start to be green and we can immediately monitor if they are activated by peptides. So in order to show how the system works, we developed library like shown here for 120 peptides, which is cancer-associated peptides and neoantigens. We clone them in K562 cells and incubate with the CMV TCR constructs and co-culture these two cell lines in Clone Explorer. And as we just definitely selected GFP positive cells, and you can see here on the right side of this slide, we identify practically in all cases CMV peptide, which illustrate power of this technology to screen different T cell receptors and peptide candidates against each other. So we believe that really functional discovery of functional TCR-Epitope interactions based on the stimulation of PBMC with peptides in vitro, it's really can be effectively performed by analysis of proliferating clone types and in combination of full-length chain pairing information using combination of bulk RSA and single-cell RSA. It's also important to really to identify activated clone types based on the expression of activation markers or cytokine secretions. But what is great, it's really in any case you need to validate these studies using cytokine secretion clonotype assay, microwave fluorospot assay, which I described in previous section of my presentation. And all these technologies are available at Cellecta if you're interested to use these technologies for your studies. And a summary of my discussion, it's we discussed development of adaptive immune or air profiling assay, which is bulk profiling assay for all 7 chains. We discuss development of single-cell air assay, which is cost-effective analysis of phenotype and full-length immune receptor chain, pairing chain information in T and B cells sorted in 96-cell plate, and with screening strategies for screening of TCR activated by synthetic peptides, vaccines, and how to validate these functional TCR antigen interactions. Thank you very much for your attention.